1, 2, 3.
Hola, Sandrita, buenos días.
Does anybody hear me? Martin here?
I can hear you, Martin. I can't hear Richard.
You don't hear? Yeah, same here. I can't hear him, and I get signs that there are network troubles. Let me check whether I can email him quickly so that we can get him back. He probably doesn't know.
Okay, thanks.
Okay.
Just stay on the line.
Can you hear me?
Oh, he's back. Okay. Now we can hear you, Richard.
Yeah, GoToMeeting does a trick on my computer where it just arbitrarily turn me off. I'm not sure what that means. Apologies. I don't know where we got lost on all this.
From me right from the beginning, Richard, but I got the sign that you might encounter network difficulties.
Okay. Just keep letting me know if anything happens, Martin.
Okay, I try. Emails or how?
Yeah, I will set it up.
Or I can try the chat.
Yeah, chat's very good. Chat's helpful.
Okay, okay. So far, so good.
All right, so what I was launching off on saying was that there are a whole lot of people doing work with networks these days, or different sites, databases promoting the concept of training networks. And it raises the question, how good are we at identifying networks and locating the important points to train within a network? And when we do it, are we getting measurable outcomes that are effective? And in order to train these networks with these more sophisticated ways, you will need very sophisticated software and very sophisticated equipment. And then we need studies to show us that this is actually better than what we're doing now. And so David Cantor and I and Dick Genardi started doing these presentations on networks. We just did one at ISNR, where we look at all the research to date on networks to try to answer these questions. And more to let you answer them for yourself. And it was very interesting to see the response at ISNR, because people ended up answering the question in what I would think would be the most obvious way. But I'll let you make up your own mind. And we'll just talk about networks. We at Newmind in our software talk about networks, but we'll talk about them in a very limited way. That there are, we identified very large-scale networks that we're just generally trying to influence. But we wouldn't pick out the network for working memory and try to find the point into it to train it specifically, down to a very, very precise point. And there's a reason for that. Hopefully that'll become apparent as we talk about networks and what's going on. And so let's just get into it. But just before we do, let's remind ourselves that single channel training has been the basis for neurofeedback and the majority of neurofeedback for decades. qEEG-driven single channel has become quite popular, and there's also qEEG 2 and 4 channel, and very much more recently, there's been some full cap training. And do those really give us an advantage, and how much? We certainly have over 250 studies showing that one channel's pretty effective. And that begs the question, can we justify more complex approaches? Is it worth it at this point, based on our knowledge base, to try to implement those, to spend money on high-level technology that might be able to implement them? And are they as efficient as what we're doing now? And do you want to spend the time and the learning curve to go out on something that's highly theoretical and very expensive and very complex? A lot of people are not really ready for that. They don't have time. They're too busy just trying to help people make a living. There are other people who have the time and resources, and they want to play with all of this a lot more. So what's the value in that? Do you want to do that? Okay. So feel free to jump in at any point if you have a comment or a question. So the questions that start to emerge are, can we identify networks at the right level? There are many levels of networks. Which one is the right level? We can look at networks in a lot of different ways. Neurons, their connections are very complex. Neurons and their dendrites bifurcate, as we say in nonlinear theory. They split like tree branches, and if you look at what they call the arborization, it looks like tree branches, roots and trees. When you look at neurons, you have this body, and out of it there's roots and trees almost. And one neuron can have as much as 14,000 connections. It gives us a really dense connectivity. That's what they call scale-free. And what does scale-free mean? There's different concepts of scale-free. There can be geometrical scale-free. There can be mathematical scale-free. Be harmonic scale-free. What we're talking about at this point is just anatomical scale-free. So the brain, the patterns of connections look the same, more or less, at every level of, when you look at it physiologically. And that makes things very confusing and complex, because it's hard to find landmarks in all of this and identify groups that are communicating. If we can identify them, these groups of networks, does the EEG have the spatial resolution we need to influence them? Just real quick, I mean, we have very good spatial resolution now with Loretta, as long as it's artifact-free. But there's problems with that too. Can we isolate specific network structures related to specific functions? Can I say, this is the working memory network over here, and this is the sequential memory network over here, and this is the short-term memory network over here, and these are their specific identities, and they are separate, and then I can influence one without influencing the other? Do we know how these networks interface, especially if they're all memory? Do we understand their spatial and temporal function? Okay. How do they behave in real time? And across what areas of the brain are they operating? Do we understand the up and downstream influences? In other words, where in the brain does processing for different kinds of memory begin? Does it begin with the images, with the emotions, with visual processing? At what point do the networks contributing each of those things come together? How do they come together? And that's the binding problem. Do we know how they come together and create a memory? And what parts of the brain are involved? In what pattern? In what sequence? In what timing? In what places? Because if you're going to go in there at a very fine point, just a few millimeters, with something like Loretta, you need to be able to hone in on these key areas, it would seem, or maybe you don't. I mean, that is a question. Can we predict the impact of a protocol if we choose a certain area? Do we know that the problem is in that area, or is it in a more complex network that's lower down or higher up in the processing chain? So I think when you start asking all these questions, it might open your mind to, well, wait a second. this might be more complicated than it looks, than just somebody has identified all the working memory network components and we're just going to go in there and train them and straighten it all out. Maybe it's not so simple. So if you have questions on this, feel free to, as we move forward. These are one of the ways that we look at brain networks in modern times. This is called graph theory. It's been evolving for a couple of decades, just about. It really started to pick up in early 2000, I think, as a way of looking at brain function and network structures. And it's based in mathematics. And this theory says that there are nodes, and you can see those words node here, and edges. A node, it might be in a network, a group of neurons, a cell assembly or something of varying sizes, and that they have fiber pathways that connect out to other groups of neurons or cell assemblies. And so the theory is that we can start analyzing the activity that we're seeing in MRIs and theorizing what areas are central and what connect to what other areas. And that's been done to some degree. So if we look at A, we can see a node, and it connects to other nodes. And if we look at that, you know, that's a pretty loose connection there. You've got just two ways of getting from that node to the other two nodes, and you have to go through them to get to the other nodes that connect to other networks. So to clarify that, in the diagram they show a network with low degree of connectivity. So you can see that one node which is having to go through a lot of other nodes in order to get to another network. And then you have in B here a high degree of connectivity where you have a central node which can get to other areas much quicker. Now the type of connectivity that is most inefficient is when every node is hooked up to every other node through an edge. So let's say this central one here was connected to all these by lines everywhere. That would require that we would have brains 3 times the size that we have now, and it would be very inefficient. So what we have identified in the brain is that there's a type of connectivity where there's lots of shortcuts. And so you have connector hubs, like the one here, and where it says module 3. So you might have a group of cell assemblies in module 1 communicating, and in a pretty tight group because they all connect closely to each other. And then you get down here another group, which is the provincial hub, and that's farther off. It's less connected. It has lots of connections within itself, but it doesn't connect directly to the other ones. It has to go through key hubs or nodes to get to the other module 1s and module 2s. And so you can see that module 3 is a connector hub. It can connect to lots of provincial hubs or other modules, and yet it's tightly connected within itself. And if it was dense enough and central enough, we might call it a major hub. Any questions on that or comments? That's kind of how we're trying to connect up neurons and cell assemblies in our minds these days. Okay, no messages from Martin. Sounds like I'm still on the air.
Sorry. Yes, you are. I hear you clearly, Richard.
Thanks, Martin. Then I'll jump over to what Sporns and Vanderheuvel have said. It should be noted that current graph-based analysis of communication cannot fully predict dynamic patterns of communication. So if you're on the internet and you see people saying, "Oh, we're processing or affecting these groups of graphs," and you'll see little Tinker Toy models, and they'll say, "Oh, this is short-term memory. This Tinker Toy model is long-term memory. This Tinker Toy model is attention." That we're going to train that area. But these guys are saying, well, factors influencing that Tinker Toy model are the dynamics of neuronal time series. And what do they mean by that? Such as the local firing rates. At what rate are all the neurons in the Tinker Toy firing? Is their activity level high or low? External inputs or task demands. What is the nature of the inputs? Because they're not just firing and not firing. There's adjustments going on with neurotransmitters to firing quality and rate. And there's phase relationships between them at their firing rates. So at what rate is one firing with respect to another firing? And what's the efficacy, generally, of the connections? Are they heavily myelinated, less myelinated, fast or slow? So those are just some of the dynamic factors that come into play when we want to say, how is that little Tinker Toy model working? There's your Tinker Toy model. So there's a dynamic, active, real-time thing going on there. Sporn says the knowledge of anatomical connectivity in the human brain is still embryonic. And compared to the current state of human connectome to a map of the world around 1570. So you can see that there's a lot we don't know if Sporns is telling us that. And he's one of the top reviewers of what's going on in the research out there in this area. So we really don't know all the answers to these questions. And we're just getting started. And yet we have these wonderful pictures. Look at this fiber pathway in the brain. And you see that picture and say, oh, well, we've got it all worked out. There's the fiber pathways. Yes, but it doesn't tell us how the Tinker Toy images map onto the fibers. And it doesn't tell us the dynamic interaction of the Tinker Toy maps that map onto the fiber. So we don't know how the networks map to the fiber pathways of the brain. And what makes this even more difficult is there's no standard definition of an edge or a node when you're looking at a group of neurons or fiber connections. Anatomical connectivity constrains functional connectivity. Yes, that we know, but we can't account for all of the rest of that stuff. Anybody want to comment? I mean, that's pretty profound thing to say about our level of knowledge, particularly when you're thinking about neurofeedback, and we're building Tinker Toy networks and saying, oh, well, we know based on DTI or Broadband areas, or we seem to be assuming we know how they all map onto there and that we're going to influence them actively. I haven't seen that research and I haven't seen anybody publish it, so it's confusing to me how people can make those claims too robustly. Freeman says to fully understand all of this stuff, we need synaptic connectivity map. And what does that mean? Well, you can dig in and find out. He's actually saying we really need clear, specific, precise maps of how all the synapses are connecting up to each other in dynamic terms. And he clearly says it doesn't exist, and DTI and DSI may never be able to produce one. So we have people out there saying, oh, we're going to use DTI or DSI to show you the Tinkertoy maps. You know, you can get a problem, and then you can figure out what problem is located to what map, and then you can train that. That doesn't seem to be a very strong possibility in these people's minds.
Quick comment, Richard.
Yeah.
Well, imagine 90 billion neurons with 14,000 connections each, and then try to map that.
Yes.
I mean, we have to keep in mind that the dimensions we are looking here, we are looking at many different dimensions. A dimension which is visible by the technological means, but that does not go to the level of neurons. So there is limitations to the possibilities we have, and a map of this number of connection is, maybe it can be produced, but it's probably not very practically useful.
Yeah,
makes sense.
Yeah, and it may not. I like what you're saying there, Martin. It absolutely makes sense for sure. And I think it's useful to try to summarize this in different ways. So I appreciate your input, because it's a big thing to wrap your mind around what's going on here.
There is another thing, you know, we have to keep in mind that if we look at network, we don't look at the data they transport. We look at the telephone connecting system, at the wires,
Yep.
not at what they are used for.
So that's the fiber pathways, right?
Even the fiber pathways is hardware, and the hardware is just the means of transport. It doesn't tell you what information is transported.
Right.
You can take the computer apart and look at it, you know, with electron microscope, but you will never find out what data processing it is doing at the moment. You just see hardware.
Exactly. And that's kind of what we're looking at with the fiber pathways, right? You opened up your computer, and you're looking at all the wires and say, oh, I see wires and I see hubs.
Yes.
But how do you know how those are programmed? How do you know how those are programmed, right?
It's a very long way to find out whether this is part of memory or this is part of motor control, data processing or whatsoever, visual processing. There's a long way between those two, the observation of fibers and what they're actually used for.
So it might be a little bit like saying, well, my Microsoft Office app is not working, and going to your computer and saying, well, if I pull on this wire or I move this one around, my Microsoft Office.
I trained this wire. I trained this wire, and then Microsoft works.
Yes.
Yeah.
I moved it, you know, or I fixed it.
Yes.
Because there's not a connection between those wires and the functionality. I mean, there is, but it's a very primitive connection. But it won't.
That's just part, you know. If one wire is broken, of course, the functionality is broken too. But if the functionality, which is observable, does not produce what we expect, it doesn't mean the wire is broken.
That's another issue, isn't it?
Yeah.
That's another issue. Yeah, that's a good point. So let's talk a little bit about how we're defining the brain. We've talked about the fact that we don't have a definition of a hub or a node that everybody agrees on, and they're all doing mathematical analyses, very sophisticated ones, based on MRI research and getting different answers. There's the question of how do you parcellate the brain? How do you define real estate in it? You know, what's the real estate arrangement? I mean, you're flying above, you know, your state, coming in for a landing in the plane, and you see all these houses and all of this farms and everything, and you kind of know that those houses and farms are part of how the real estate is defined, but you can't see the county lines drawn. You can't see the property lines drawn. Sometimes there's a wall, sometimes there's not. Sometimes it's a river, sometimes it's not. So you don't really know how things are broken down. You can just say somehow these people have boundaries, and we don't know exactly which ones are which. Do two of the farmhouses belong to the same family? How much of downtown is owned by one person? How is things divided up? We don't really know that. What's the best way to look down there and make a decision? Do we make it based on houses? Do we theorize about the stone walls? Or do we theorize and say, well, they might have a dynamic, non-material way of dividing this up in the same way that software is non-material. What people are looking at in terms of different methods is interesting. I mean, they're using Brodmann areas in one set of methods, but they're also using other, and we don't need to get into all of them. There are other ways of breaking the brain down into areas functionally besides Brodmann. And there are people contending that the Brodmann area is not the best way to do this. So there's a disagreement about that. Well, that hasn't bothered us in neurofeedback. We just assume that the Brodmann area is the way to break it down a lot of the time, but it might not be. So Dick Genardi weighed into all of this. This is one of his PowerPoint. And he said many imaging modalities in identifying nodes use different measures. PET, metabolic, blood, MRI, fMRI, right? Spectral power, EEG, MEG, MEG, electromagnetic readings. So we're identifying nodes using different measures. But the time domain of the MRI is several seconds, whereas EEG is milliseconds. So we're going to get fundamentally different information on important nodes looking at MRI and EEG. Hence, different resolution time different resolution due to time dampening. We do know that you can roughly correlate MRI with EEG, but we don't know the details yet. The correlation of heightened activity when somebody's doing a task, reading, writing, whatever, of a node identified by one modality may not fully understand how it relates to node identity by another modality. And there's whole articles like Zaleski, and I read that article. You read that article, it's you realize how deep this gets, that disagreement on what's a node and a hub and why, because people are using different rulers to measure these things. So one person's got a meter ruler, another person's got a Sumerian ruler, another person's got a centimeter, a stick. Some people may just be guessing. So a network is not simply defined by co-activating nodes. You can have two things activating at the same time, but they may not be connected. You have to show that they're connected, just like in causality theory in research. There needs to be a connection between two things occurring at the same time, or they may not be related at all. And how do you confirm that? And then there's a use of different values for parameters, which results in different nodes and connectivity patterns. So those are some of the complications with saying, okay, how do we carve up the real estate here? What imaging technology has the best handle on what the real estate's doing? And how do we connect things like thoughts and actions and feelings and perceptions to the wiring, the hardware, the hubs and the nodes? Most anatomical parcellation studies have focused on the cerebral cortex. Less attention has been paid to subcortical structures such as the basal ganglia and the thalamus, which have only been demarcated at a coarse level using MRI. Brainstem systems mediating motivation, autonomic function, and arousal have been poorly studied because they are notoriously difficult to identify using in vivo techniques, getting in there when it's really happening, when somebody's alive. Nonetheless, it's important to identify these structures because they significantly influence cortical signaling and thus affect cognitive function. So, okay, let's say we have some idea of what the large-scale networks are, but as I've said in the past, they can't function properly if the affective system isn't working right. If our arousal levels are not correct, the brain can't work right, you know, your cognitive functions. If you're too emotionally disturbed, your cognitive faculties can't work right. The whole system phylogenetically built upon a hierarchy where those are ascendant. And so when we're in fight or flight, all the top stories get shut down. The cognitive system is suddenly not even there for the most part. So, you know, we have direct proof of hierarchy and level of importance, and yet we don't understand these substructures very well. So, okay, you've identified as what you think is a network brain, and maybe you're not. Somebody was yawning there. Okay. Any more comments at this point? Input? Okay, let's talk about levels of analysis in the scale-free system. We've got micro, meso, and macro. Micro is the smallest level, down to just a few cell assemblies, groups of cells. Meso is in between. That's more the where all of these different cell groups are co-activating. And so in EEG terms, we'd be talking about a dipole layer, according to Nunez. And that dipole layer is resonating between all of the different cell columns and neurons in those cell columns, and we get a large enough area resonating in a specific frequency, 60 million or so, we can get a signal at the top of the brain. So we have to look at that. And then we have what we call the large-scale networks at the macro level, if we're thinking in terms of network analysis. But we can also think in terms of wiring and layers in the brain. We're not really sure how that hierarchy works. First, current large-scale models of communication in the human brain capture only interregional projections, accounting for only a small proportion of neural activity, and do not include networks of local circuits. So when we're looking at the big scale stuff, we're just looking at the final outcome, and all of those millions of neurons and the thousands, perhaps hundreds of thousands of networks with hubs and nodes that are talking to each other, firing at different rates, we can't say. We don't know. We can't measure. There's too much. Local processing of neural signals is undoubtedly an important aspect of brain communication because it involves the transformation and recording of neural messages at each node of the network. So we need to know what's going down at those levels if we're really going to understand a node or a network. And I've shown this slide quite often. Phase synchrony of neurons determines amplitude of EEG activity. We know that neurons just firing at random don't produce anything that can be read at the scalp. But if we have patterns, sequences of firing, the dynamic quality that's based upon the connections, if that is non-random, and there's enough of them all doing the same thing at the same time, they're synchronized, then we can get energy signals on the scalp. So 60 million neurons, perhaps an alpha, all doing alpha at the same time. And and the timing is just right so that each one is firing close to the same time and not at a different time. Get 60 million of them, we can get in that region of the brain, approximately 6 square centimeters, we can get a signal on the scalp. So that's pretty vague if you think about it. It's not very specific, and with EEG we're trying to sort that out. And we don't really understand the relationship between delta, theta, alpha, and beta, and which of these networks are producing them at which time and why they're doing it. For instance, delta is very much a frequency that connects the brain stem with the frontal region of the brain and is critical for salience processing, looking at something and determining whether it's something of value. And it connects up through limbic structures and arousal structures, going from the frontal areas down deep into the brain. And those areas have to all be synchronized, but we don't know why the cortex harmonics are occurring the way they are unless we also include the story of what's going on in the subcortical areas. And we just said we don't really have that worked out very well. Okay, we need to look at more at the spatial scale limitations of EEG. As we said, dipole layer size is a function of the amount of synchronous activity, the size of the area of activity, and the synchronous cell distribution in the dipole layer. Dipole layers must be at least 6 centimeters squared in size to be recorded at an electrode. This is approximately 60 million neurons, and they must be synchronously occurring. You can get an idea about half of the inferior frontal gyrus is that size. So with EEG, we're getting pretty broad signals. Now we can, with Loretta, get down to very small sizes, 6 millimeter cubes, voxel regions. But then when we get down to that size, we don't really understand still how one group of cells is talking to another group of cells and how far that discussion extends from one Brodmann area into another. And keep that in mind because we're going to show you research later showing that Brodmann areas are not by any means homogeneous, and that when you're looking at imaging in one, which real estate in there is the real estate you want to be looking at. Now Thatcher has said, he said he's done the work, and if you look at, just pick the center of these things, based on if I'm reading him correctly, and that you look at all the other areas and you average it out, the center is a good estimation of the general average of the activity in the rest of the area. Well, that may be so statistically, but those other areas may be waxing and waning in ways that are disconnected. So again, we have co-activation, but not necessarily connected. So does that average really give us the information we need? And if we look at the cells and the cell columns, we can see, I think this is a nice picture. I always like to show this. Because you can see the fibers. These are thalamocortical fibers only. There's fibers between the cells, but we also have the thalamocortical. Look how densely those are connected to the cell columns. And then there's your different layers of different cells with slightly different functions. And we don't really know how this all connects up with processing, with networks, with the dynamic networks. So here are some of the terms that we've been using to describe this spatiotemporal orchestration of neurons. And you've heard me say cell assembly, just small cohorts of cells networking together for 30 milliseconds or more. That's kind of the fastest you can do this kind of stuff in the brain. Then there's synaptic action fields. That's the number of excitatory, inhibitory synapses per unit volume. That's independent of participation in cell assemblies.
Richard, question.
Yeah.
To the cell assemblies, 30 milliseconds, has that been established because measuring equipment can't go faster, or is that what the brain maximum can produce in terms of speed?
Based on Nunez's calculations, that seems to be the fastest we can go when we're looking at electrical, current.
To produce a column strong enough for a group
of cells. That's the fastest they can get together in terms of the physics of how everything works. But that could be wrong if there's a quantum component to it.
Yeah.
And
thanks for clarifying.
Yeah, and there's evidence that there may be a quantum component. I mean, if you're kind of glazing over or reeling with all of these concepts, it's not a bad thing. You're starting to get a real picture of how complicated these are and how much everybody's struggling and how it contrasts with how simply everybody wants to present it in neurofeedback sometimes.
These things happen at the molecular level, neurotransmitter activity and so forth. So quantum component makes sense on that level. It doesn't on an organism level, but on a molecular dimension, it starts to make sense.
It does. There's a efficacy recent articles proposing that the actual cytoskeleton structure of the cells is not structural, but it's the cell nervous system, and it's related to the same filaments that microtubules use for their genetic activity, and that those fibers actually resonate at quantum levels. So,
Bruce Lipton came to that conclusion as well, not with neurons, but with other body cells, some 30 years ago.
Mm-hmm. Well, there's strong evidence for these fibers resonating at those frequencies. So, to make all of this worse, we may be only looking at, 2/3 of the picture. There may be a third of it, may be all quantum functions. Okay, and then we have the microcolumns, the actual physical grouping of the cells and their fiber pathways, and we've tracked a lot of that down, but they all talk independently within and between cell columns. So remember, this one neuron can get to any other neuron by going through fewer than 3 neurons, is the rough estimate for all these massive amounts of neurons. Then you hear a lot of talk about regions of interest, which is a Brodmann area perhaps. But the Brodmann areas have multiple functions many times. So which section of the Brodmann area is related to which function, or can they even be parceled out? More recently, MRI activation studies have been used to more precisely demarcate nodes of specific functional circuits associated with dedicated networks. So those are the ROIs. However, regions of interest identified in this manner tend to vary considerably with task demands, patient groups used, and the specific control or baseline conditions used to identify them. As a result, uncovering the nodes of neurocognitive networks in a principled and reliable manner has turned out to be elusive. We have problems with identification, problems with measurement, and problems in real time with their stability, because they can seem like a region of interest is associated with one task ability when it may be actually part of another group, or other groups may be part of it, and that its function changes depending on subtle changes in task. And then we
Check
have
the time, Richard. We are getting close and
Yep.
might lose some of the audience.
Okay. Yes, we are. So this is getting close to this time. So we're just getting into this, and so it's probably a good time to drop off here. And where we're dropping off is that not only is communication between fiber pathways, it's also chemically between neurons. It's also harmonically between neurons. So we have multiple levels of communication that are not fiber pathway based. And these harmonic connections we deal with a lot in neurofeedback because the connections between theta and gamma and beta and alpha and beta. So we'll talk about that next time. Talk about comodulation, these harmonic influences maybe. So hopefully that at least got you some insight into the complexity and some of the concepts and terms used. You may be familiar or not familiar with them, and gets you thinking about it a little bit so that when people talk about it, you can listen more critically. If anybody have any closing comments or concerns, we'll pick this up on Friday.
No, just thank you very much, Richard. This is fascinating stuff.
Yeah, it is. It is. It's overwhelming, isn't it? Okay. So Friday, we'll talk a little bit more about this, and this will probably relate a little bit more to neurofeedback because we'll be talking about the relationship between the component bands more. So have a good week, and we'll see you Friday at noon.
Thank you, Richard.
Yep. Thanks, Martin.